Hi there, science fans. Welcome to another episode of Science and Other Stuff for Senior Citizens. My name is Dan Casciano. In the present video, I will describe several new technologies used in the development of vaccines to neutralize the Covid-19 virus. One of these technologies already is in use to facilitate herd immunity and to eliminate the Covid-19 pandemic. First, we have to review some immunology. Prior to the onset of the Covid-19 pandemic, the majority of our vaccines were developed using proteins or small peptides that were associated with the invading foreign organism. In cartoon A on the left, several vaccines in development contain pieces of the Covid-19 spike protein that we now know elicit production of antibodies to the spike proteins in COVID-infected humans. The virus's spike protein is the structure that interacts with the surface of a human cell, resulting in entry into the human cell. Upon entry into the cell, the virus makes many more viruses to infect other nearby cells as well as infecting other humans. Production of the spike protein antibodies by the immune system results in inhibiting the ability of the virus to penetrate nearby cells. Proteins or peptides that induce production of antibodies are called antigens. Cartoon B on the right represents how the newly developed mRNA vaccines do the same thing. These vaccines also induce the formation of antibodies to the spike protein, thereby neutralizing further infection by the virus. The messenger RNA contains the information necessary to form the spike protein antigens that are responsible for the induction of antibodies to this antigen. The new techniques discussed in this video are nanoparticle vaccine delivery devices and CRISPR-Cas9, which is a DNA editing system originally discovered in bacteria with incredible applications in the human and in other species. A nanoparticle is a particle or molecule of an extremely small size. As can be seen on the right side of this cartoon, the nanoscale generally includes 1 nanometer to 100 nanometer objects, but can also include atoms that are measured using angstroms. A glucose or sugar molecule is around 1 nanometer in size, while a virus like HIV is around 100 nanometers. In addition to use in commercial products, nanoparticles or nanodevices are created to interact with cells and tissues to carry out very specific tasks, such as treating disease and preventing health issues. These latter uses have resulted in the development of nanomedicine, a branch of medicine that focuses on the development of nanodevices that can target specific disease cells and tissues while leaving normal cells and tissues intact. The nanoparticles used during the present pandemic include a lipid nanoparticle encapsulated mRNA, as well as an inactive common cold adenovirus. The Pfizer and Moderna vaccines use a lipid encapsulated mRNA. In this case, the lipid is an oil droplet, while the AstraZeneca and the Johnson & Johnson vaccines use an inactivated double-stranded DNA virus called an adenovirus. In some of the gene therapy cases undergoing human clinical trials, adenoviruses are used as carriers for molecules to treat the specific genetic disease under consideration. Now that we have several vaccines against COVID-19, questions arise whether these vaccines will work on mutants or variants of this virus, and if these vaccines will protect us from other strains of the coronavirus. Research recently published in the journal Science is investigating the potential to train the immune system to produce antibodies to multiple strains of coronaviruses with a single injection of a vaccine. This research is based on technology called a mosaic vaccine platform that is represented visually in the cartoon on the left. The central cage-like nanoparticles has attached to it several different spike protein fragments, or antigens called antigen A through antigen J. When a system like this was injected into mice, a model system used for work in immunology, there was a diverse antibody response such as that depicted in the cartoon on the right side of the slide. The initial results are encouraging because the mice began producing antibodies that could neutralize many different strains of coronavirus. While studies like this are still in their infancy, the potential for applications in the human is enormous. The 2020 Nobel Prize in Chemistry was awarded to Emmanuel Charpentier and Jennifer Doudna for their groundbreaking work on the discovery of the DNA editing tool CRISPR-Cas9. Their revolutionary work on cutting DNA where you want allows editing the DNA for removal of deleterious information and the addition of beneficial information. One application of the CRISPR technology may be for the rapid development of antibodies to variants or mutants of COVID-19 viruses that have escaped neutralization by our present vaccines. Site-specific modification of antibodies by site-specific modification of targeted sequences in the genome could revolutionize the field of immunology. These areas of research are also in its infancy, and its potential application to the compromised human is enormous. The emerging applications of the CRISPR DNA editing technology is still in its infancy and has so many applications to the human. Because of its complexity and the requirement to clearly explain its utility, I will be dedicating several videos in the near future to this technology. Those of you who would like a glimmer into its mechanism and potential, I've included several YouTube links in this slide. Here is some bonus information. Johnson & Johnson has completed a human clinical trial investigating the safety and efficacy of the vaccine it is developing and will request the FDA for emergency authorization for human use in the first week of February. Although the efficacy is not quite equivalent to the Pfizer and Moderna vaccines, it is more than adequate to neutralize the COVID-19 virus in infected patients. As far as I know, it is the only vaccine shown to protect patients from severe disease outcomes. This means many old people and many people with health issues. Although vaccines are beginning to be administered to humans, we must continue to clean or wash our hands often, avoid close contact with sick people, practice distancing 6 to 10 feet, avoid indoor and outdoor crowds, and wear a mask properly. We will continue following these CDC guidelines until we reach herd immunity. For more information about science, visit my website at www.dcasciano.com. If you have any questions or would like me to develop a video on a specific science subject, leave them in the comments section below or email me at dan@dcasciano.com. Be sure to like this video and subscribe to my channel and ring the bell so you can be informed when I upload a new video. Thank you for watching Science and Other Stuff for Senior Citizens. Bye.